I am presenting on June 6, 2026. Auto transplants treat patients with chronic pancreatitis. A surgical team removes the diseased pancreas at the designated medical center and transports it to a clean room facility where a team performs benchtop surgical work on the pancreas to clear away the junk surrounding organs like spleen and duodenum, fat, fibrotic tissue, blood vessels, etc. Dissect it to cannulate and inject the ducts with an enzyme mixture to break down the pancreas while releasing the islets. Collect as many islets as possible through the Recordi chamber, a specialized device containing a filter inside that separates islets from the remaining junk, and prepare IV bags of islets to send back to the patient where the islets are infused into the liver portal vein for engraftment. That way, the patient can still produce insulin when needed without the need for pumps and without any inflammation. For islet allotransplantation, the steps are the same, except that this time the pancreas is coming from a healthy deceased donor, and this procedure typically treats type 1 diabetics. Once the pancreas is removed, the team has 12 hours to process it before it begins to digest itself and the islets. So the earlier the organ arrives, the better the islet yield will be following collection and infusion. The reason type 1 diabetic pancreases are not good for auto-islet transplants are due to the following reasons. The extracellular matrix is abnormal to the point where it's harder to break down the pancreas and it's trapping the islets. The organ is a smaller size, likely due to the hypoxic environment created by the autoimmunity and inflammation, and the islets lack beta cells that produce insulin. The following dilemma is that the FDA classifies islets for allotransplants as a biologic drug. In Section 361, the product is considered an organ or tissue if it was minimally manipulated and was for homologous use, such as replacing like with like tissue. While in Section 351, the product is considered a biologic drug if the tissue was more processed than minimally manipulated and was not for homologous use, altering the tissue's biological trait for new use. The islets for auto-islet transplants are not considered biologic drugs, while the islets for allotransplants are, despite that the steps of processing them are the same. And the purpose of these transplants are to ensure that the patient is still producing insulin when needed, just like how we would with the pancreas. The reality seen during these cases is that every pancreas is different. The number of islets collected per case are different from other cases, or if islets were a drug, which amount is correct to ensure that the patient has good recovery. What if there weren't enough islets collected and the process is delayed due to waiting for the next available donor? What if that led to a shortage in donor organs? If islets were a drug, that may enable pharmaceutical companies to make the islets for profit, leading to an increase in the cost of transplant, as well as limiting access to the treatment for patients. It's 1.2 million of them in the US who need it but cannot afford it. Because the FDA classified islets for allotransplants as biologic drugs, they are requiring transplant centers to receive an approval of a biological license application or BLA before clinical use. This requirement stalled 20 years of development in the field, and a private for-profit group, Celltrans, submitted theirs and recently it was approved. which will allow commercialization of islets for transplantation. Granting the BLA to that group is consistent with the prohibition of the sales of human organs. It's a criminal offense to sell a kidney according to the National Organ Transplant Act. The only exceptions for sale will be bones, skin derivatives, blood, plasma, sperm, and eggs. This law also created the Organ Procurement and Transplantation Network, or OPTN, operated by United Network for Organ Sharing, or UNOS, to oversee organ matching for allotransplantation. Because the FDA classifies islets for allotransplants as a biologic drug instead of an organ, there will be no oversight on the islets, which may jeopardize the safety and effectiveness of the allotransplants if the BLA was approved. Following the approval, the group will have 7 years of marketing exclusivity under the Orphan Drug Act of U.S. law from 1983 that incentivizes pharmaceutical companies to research and develop treatments for rare diseases. Significant leverage in terms of the contract with any transplant center to provide islets that comes with a cost. An unprecedented influence over which transplant centers can offer the treatment. Overall, transplant centers will have no alternative source of islets for clinical use, and consequently, there might be shortages and there will be no control over quality of the islets for these patients. Transplant teams already follow all kinds of protocols to prevent contamination and take multiple sterility tests to send over to a microbiology lab. They also check whether the islets are freed or trapped during collections. Um, Purify the islets if the pellet size is large. Check viability. Do gram staining to check for clinical contaminations. And do cell counting to determine how many IV bags to prepare to send to the hospitals. Lastly, access to the treatment will be limited due to the increase in cost and decrease in availability of the islets. BLA can compromise patient safety. Under the Health Resources and Services Administration, the OPTN and UNO's oversight of the transplants is critical to assure the safety and effectiveness of the treatment. With BLA, islets and patients will be excluded from this oversight and post-transplant monitoring. So ironically, the islets will be less regulated. The weirder thing is that the FDA's position on BLA for allotransplants is inconsistent with their approach to auto allotransplants, despite that steps of processing are the same. At least 50 islet experts and endocrinologists classify islets as microorgans and should be treated as any human organ, which is not regulated by the FDA and not BLA required for the following reasons. Islets are not artificially manufactured. They obviously exist in the human body, and it's not like a lab synthesized them from chemicals or extracted and purified an active compound from a plant. Islets are a cluster of cells with their own functions, and they have their own circulatory and neural network. They maintain their own morphology during islet isolations and following transplantation. They connect their own vasculature to the recipient's liver portal vein for engraftment and in order to function properly like they would in a pancreas. Islets can't handle below zero temperatures and are only preserved at room temperature for a short period, making it impossible to have islet cell banks. And unlike drugs, the outcome of the allotransplant depend on the following parameters in contrast to a single test. The sterility of the environment the processing takes place, the sterility of the team members, any minor nicks in the pancreas during processing, and constant supervisions of donor selections, checking for damages to the pancreas, which may happen if the surgical team is harvesting blood vessels on top of donor availability and the OR time for the recipient. And that'll coordinate it with the donors. Supervisions of the processing with documentations and reports to share with the transplant team, infusion, and post-transplant care. The only way to know potency of islets is not through a certificate, but with how well the patient responds after transplants. Lastly, as mentioned before, every pancreas is different. So, how can they be standardized as drugs? Some academic centers have been able to perform allo-transplants without the BLA for the past 20 years. Their labs are not drug manufacturing centers, so what's the point of having a BLA? After those 20 years, islet allotransplantation is not broadly available to American type 1 diabetics, while in other parts of the world, islets are classified as organ/tissue, and the treatment is more available as a standard of care. Ironically, these worldly programs benefited from the American-led research behind the development of this field. There are issues seen from a consequential view of the BLA approval, where it's inconsistent with the laws of prohibiting commercial sales of organs, and it jeopardizes the patient outcomes regarding control over the quality of islets and post-transplant monitoring from OPTN and UNOS. It also limits the patient's access to the treatment, and it may jeopardize with jobs of members of the transplant teams. From a utilitarian approach, more American type 1 diabetics will be able to get allo transplants without the need of transplant centers sponsoring a BLA and without monopolization and increasing costs of islets for the procedure. From a deontological approach, classifying islets as biologic drugs will limit patient access and jeopardize their outcomes after the procedure by limiting the oversights. The autonomy is going to be limited due to companies like Celltrans dictating which transplant centers offer the treatment for whatever costs they come up with, and what providers are allowed to treat their patients with, further limiting their access. There's a lack of beneficence due to limited access and safety of the treatment. The BLA approval and FDA's definition of islets for allotransplants fail to follow the non-maleficence and justice principles by preventing beneficial therapy for those who need them. Islets, of course, should be free of contamination. When these companies follow the current good manufacturing practices, but patients are still exposed to uncontrolled clinical risks. By applying the fidelity principle, islets naturally vary, especially the number of islets per pancreas, and cannot be standardized as drugs. Celltrans did their submission to ensure the safety of islets for transplants, even though there are quality checks of the islets during auto islet transplants. So once the islets are expected to be regulated as biologic drugs, companies like Celltrans are also going to be strict on purity and potency of the islets. Hence, they'll be required to check viability and perform glucose stimulation, insulin secretion to check the islet functions before releasing it to transplant centers. The other is to consider the risk assessments, given that patients will undergo immunosuppression prior to and after allotransplants to prevent graft versus host disease. The FDA argued that islets for allotransplants are not analogous to whole pancreas transplants, so far the only kind of transplants for type 1 diabetics. By recalling how the organ decreases in size from the disease itself. There are cases of newer pancreases getting smaller due to the patient's hypoxic microenvironment and leading to abnormalities in the extracellular matrix, just like what's been occurring in the disease. While Cell-Trans intended to optimize the safety of the islets, the viability changes over time. It's not possible to have a form of an islet cell bank due to viability and due to their fragility at below zero temperatures and room temperature. The other part of the problem will be the delays to check islet function through glucose stimulation insulin secretion assays. Are the islets going to be hand-picked and put into round bottom wells to form aggregates? Or are they going to undergo dynamic parafusion? The supernatant collective will also have to undergo an insulin ELISA to measure insulin secretions and to calculate the glucose stimulation index in order to determine whether the islets are functioning properly. Also, in order to match the donors to the patients, Cell-Trans will then have to be responsible for screening the donors, their age, BMI, and any underlying conditions, addictions, and serologies to determine if the donor had any infections, further limiting patient access due to the number of pancreases that were accepted. While islets for allotransplants are not analogous to the whole pancreas transplants, they are microorgans due to being clusters of different cell types. Alpha cells to secrete glucagon when you're fasting, beta cells to secrete insulin when you're eating, delta cells to produce somatostatin to help keep blood glucose levels balanced, neural networks to induce calcium channel activities to induce hormonal secretions into the microvasculature. A lab cannot produce islets like they can produce drugs because islets exist naturally in a human body. And while the FDA is concerned about how much tissue manipulation occurs during islet processing for allotransplants, the steps for the processing the islets are the same for auto islet transplants. The only difference is where the pancreas came from. In other words, islets are islets. Therefore, I and many islet experts suggest that the FDA should reconsider their illogical classification of islets for allotransplants as biologic drugs and the requirement for transplant centers to sponsor a BLA and instead think about the many people who desperately need this treatment, especially without financial barriers and without the fear of their safety and well-being after the transplant.